Hello students, in previous part we were studying some homopolysaccharides such like as starch and cellulose. In this part we will discuss on homopolysaccharide, heteropolysaccharide and amino acid. Homopolysaccharide, which we have already studied. Homopolysaccharide means the carbohydrate long chain which is made up of one type of monosaccharide, that is called homopolysaccharide. We have already studied about cellulose and starch. One more, number third, one more homopolysaccharide is glycogen. Glycogen is also homopolysaccharide which contain many units of glucose. Glycogen is the storage starch in animal. It functions it functions to store starch in animals. कहाँ पे स्टोर करता है? इन लीवर एंड muscles. This is the function of glycogen. Glycogen is homopolysaccharide which contains many units of glucose. It shows functions to store starch in animals, in liver as well as in muscles. Glycogen form 1,4 glycosidic bond. Which type of 1,4 glycosidic bond? Alpha. Alpha 1,4 glycosidic bond and many 1,6 glycosidic bond. So we can say that glycogen show linear structure as well as branch structure. Alpha and beta, what do you mean by alpha and beta glycosidic bond? We have seen that in glucose. In glucose, one carbon molecule, or we can say that one glucose molecule attached with the other one, so this one, this is the carbon number 1, 2, 3, 4, 5 and 6 remain here, and this may contain carbon number this may contain carbon number 1, 2, 3, 4, and if this structure may remain as such with this one 4 and 5, 6 remain here. Listen, the glycosidic bond 1,4 beta and alpha. What is the difference between beta and alpha? If carbon number 1 contain OH group upper side and bonded with 4 that contains loss of water, H and OH. If carbon number 1 contain OH group upper side, then it forms alpha bond. If carbon number 1 contain OH hydroxyl group lower side, then it will forms beta glycosidic bond. This is the major difference between alpha and beta glycosidic bond. The next one is, what is the feature next or other features of glycogen? Glycogen represent red color with iodine. Glycogen represent red color with iodine. As we have studied in last previous part, starch represent blue-black color with iodine solution, but glycogen show red color with iodine. The next one is chitin. Chitin, it is also a homopolysaccharide. It is the homopolysaccharide of N-acetyl N-acetyl glucosamine or amine. It is the more chain of N-acetyl glucosamine and form a chitin. What is the function of chitin? Chitin represent the structural formula or structure of cell wall in fungi as well as exoskeletal system in arthropods. Structural, it helps in the formation of structure. Cell wall in fungi and exoskeletal system in arthropoda. Chitin is also homopolysaccharide which contain a long chain of N-acetyl glucosamine. And what is the function of chitin? It helps to make the exoskeleton in arthropoda as well as cell wall in fungi. One more homopolysaccharide is Callose. Callose is also homopolysaccharide. Homopolysaccharide of glucose. It means it contains a long chain of glucose molecules. In Callose, 1,3 glycosidic bond formation can take place. 1,3 glycosidic, it means one molecule of carbon number 1 of glucose combined or bonded with the other glucose molecular carbon number 3, that is the Callose. What is the role of Callose? Callose formed in pollen tube, just like a formation of block band. As the male germ cell pass out, pollen tubes is found in style. As the pollen grain fall off on stigma, they start to germinate and form a pollen tube. As the pollen tube forms, there are two male gametes or germ cells are present. They contain cytoplasm. These germ cells, when they pass out through lower side, the callose forms block band. It means they help to pass the germ cell from one side to one place to another place. They form the block band, and this block band is formation can take by callose. The other one is, we know that in pollen grains. Pollen grains, there are two types of cell. One cell that is large inside, that is called vegetative cell, and other one is known as a germinative cell. This is the nucleus, germ cell. This is the germ cell that is small in pollen grain. The cell wall of germ cell is also made up of callose. The cell wall of germ cell or germinative cell is made up of callose. These are the some important homopolysaccharides. There are some other homopolysaccharides, inulin, agar, there are many. And next one is heteropolysaccharide. What is heteropolysaccharide? As the name show, the polysaccharide which is made up of different types of monosaccharide chain. Heteropolysaccharide. The carbohydrate which contain many types of monosaccharide and form a long chain. This type of carbohydrate is made up of different types of monosaccharide chain. The main example of heteropolysaccharide is, for example, hemicellulose. Hemicellulose. Hemicellulose is made up of xylose, arbinose, mannose, and galactose. And galactose. Hemicellulose is the example of heteropolysaccharide which contain many monosaccharide of xylose, arbinose, mannose, and galactose. They contain a long chain of different types of these biomolecules. So hemicellulose is made up of, is known as heteropolysaccharide, where it is uses the use of hemicellulose in the formation of or production of alcohol by the fermentation process. It is used in production of alcohol by fermentation process. These are the polysaccharide biomolecules. The next biomolecule is amino acid. Amino acid, which are known as the protein synthesize. Amino acid. There are different types of amino acid present in our body or synthesized in our body. But out of many, only 20 amino acids synthesize the protein and more than 100 perform different functions. 20 amino acid synthesize the protein only, and more than 100 perform other function. So we can say that there are different types of amino acid present in our body. Amino acid is the group of methane which contain one amine group and minimum one amine group and one carboxylic group. Methane, we have already studied about methane. Methane is the one carbon molecule which is bonded with four-side hydrogen molecule. As the amino acid formation can take place in this methane group with carbon one side attached with the amino group, other side with carboxylic group, and one side with H. The fourth side remains here with another molecules, which makes the different types of amino acids. Such we will discuss later. This is the group of amino acid, the group of methane group. Just like as methane, it is the carbon molecule is bonded with the four side, so it is a saturated. In amino acid, one side of carbon is attached with the amine group, minimum one amine group and one carboxylic. Amine group is basic in nature and carboxylic is the acidic in nature. The presence of amine and carboxylic group which show the nature of amino acid. How many amine group or carboxylic acid groups are present in the amino acid? That show the nature of the amino acid. For example, if the amino acid which contain number of amine group is equal to the number of carboxylic group, then this type of amino acid is neutral. This is the neutral. For example, glycine and serine. Glycine and serine which contain equal number of amine group and equal number of carboxylic group. One more type, if the number of amino group is higher than the number of carboxylic group, then this type of amino group is basic in nature, and that is basic in nature. For example, lysine and arginine these are the basic amino acid. But if the number of amine group is less than the carboxylic group, amine group is less than carboxylic group, then this type of amino acid is acidic in nature. Acidic in nature. For example, glutamic acid or aspartic acid. These both are acidic in nature. So, the presence of number of amine and carboxylic show the nature of the amino acid group. The next one is, there are, on the basis of requirement, amino acid is of two types. On the basis of requirement of the body, the amino acid is two types. Types of amino acid on the basis of requirement. Two types, essential amino acid and a non-essential amino acid. Essential amino acid and non-essential amino acid. Non-essential amino acid. Essential amino acids are those acids which cannot be synthesized by our body. They must be taken through our diet. These are taken through diet, or we have these type of amino acid in our diet. They cannot be synthesized by our body. These type of amino acids are known as essential amino acid. For example, tryptophan, methionine, phenylalanine, these are the amino acids which cannot be synthesized by our body, but they are very must be required to the body. Non-essential amino acid, those amino acids which are synthesized by our body. Requirement is also essential, but they cannot be taken through outside. They must be synthesized our body. The amino acid which synthesized by our body. For example, glutamic acid, aspartic acid, they are synthesized by our body on us. A few amino acids which are known as semi-essential amino acid. It means these amino acids are required only some special function, not all the time. These type of amino acid, these types of amino acid which required only at the time of growth or function. When the cell performs special functions and the cell show growth, these amino acids are to be required. So they are known as semi-essential amino acid. For example, arginine and histidine. Amino acid, we can say that the example of ornithine, histamine, more different types of amino acids are present in our body. Clear? How is the structural formula can take place in amino acid? As we have discussed, amino acid is the group of methane group. Just like as one carbon molecule attached with the other side with amino group and one side with carboxylic group, third side will remain H and one side remains the another factor. This another factor show the different types of amino acid. If at the position of R, it may remain as H. For example, such like this one side carboxylic, one side N amino group, other one is H. But at position R, if it also remains attached with H, this is simple amino acid. Simple amino acid, that is called glycine. Glycine. This is the formula of glycine. But in place of this R factor, if it is attached with methyl group, such like as one carbon molecule attached with amino group one side, another with carboxylic group, one side remains H. At position R, if here is one methyl group is attached, this type of amino group is known as alanine. Alanine amino acid. And when at position R, if here attached with the CH2OH, then that amino acid is called serine. For example, this one C, one side attached with amino group, other with carboxylic group, one side with H, and position R, if here is attached CH2OH, then this type of amino acid is known as serine. Which one amino acid? Serine. So we can say that on position R, what will be the attached with the methyl group, or we can say the methane group of amino acid? That show the different types of amino acid. The next one is, what do you mean by polypeptide bond? Polypeptide bond. The bond which is formed on losing of water molecules and attached between, or we can say the formation between the ketone group and NH. For example, polypeptide bond. The bond which is formed between CONH means ketone with NH group with losing of water molecule. As the amino acid form a long chain, during the formation of long chain, polypeptide is formed. For example, we can take the example of two amino acid example. C, NH, one side of amino group, one side with H, other side with R, and it attached with the COOH, one side with carboxylic acid. This is the simple, maybe any types of amino acid. When it is attached with the other one, carbon, it contains H, N, it contains H and H, NH2, same amino group. One side with R and other with COOH. During this formation, one OH from here and H from here, lost water molecule, H2O, lost water molecule, and it will form between CNH2H R, and this may remains only CO. Other will remain same as CH, this side R, COO, and this side it remains only NH, because this type of OH and H lost the water molecules. It may contains NH. Here is the bond formation can take place. This bond is known as a polypeptide bond. So we can see that what do you mean by polypeptide bond? The bond which is found between the ketone group or NH group. While formation a long chain of amino acid losing of one water molecule, that type of polypeptide, that type bond is known as polypeptide bond. Next time we will discuss of another type of biomolecule.